AC Muscarine. Perfect. Okay, so the next exercise, we're going to be looking at gene models and assessing whether they're kind of correct or not, and kind of look at it more subjectively based on, say, the evidence that we have in our database. And for this exercise, we're going to be looking at ToxoDB, and the first gene we're going to look at is a fancy one, is hyperxanthine phosphoribosal transferase. That's a bit of a mouthful, but this was the gene in Build 65. And this is not what's currently shown on the website, but I will go through that shortly. So, is this right? I mean, at the moment, we've got one gene on a page. I can't really tell. There's no evidence. And so, this is what you need to make correct inferences about the gene. You need evidence, right? And so, what we've seen from Gloria and Omar's talks is that we do have a lot of data available in our database to make these assertions. And so this is Build 65, and our wonderful curator who's on the call right now, Uli, she's created 3 other isoforms of this gene, and that has been loaded in the previous release. And so we asked the question, this is better? Question mark. Can we do better? I mean, is there any evidence that we can use to kind of justify maybe deleting an isoform? Maybe Uli got it a bit wrong, or maybe adding an isoform just because different life cycle stages, this gene is in different exonic order. And also, we changed the name of it, so it's now hyperxanthine xanthine guanine phosphoribosal transferase, which is an even bigger mouthful. So, in the exercise, you'll get a link to the next slide, which is quite a few different configured tracks that we can look at for evidence to assess this gene model. So currently displayed, we have the consensus gene set. So this is the gene set for this release. Underneath it, we've got the Build 65. We have transcription start sites. We have the intron spanning reads. We have multiple other evidences, which I will go over now in a bit more detail. So these 2 models, as I said before, release 65 track, the colors are a different set than the red and the blue from that are currently there. And as we can see, we've got the 4 different isoforms here, and in the previous release, just the 1. We also have a community annotations track, and I believe Uli will talk about that later today, which is where you can create your own in Apollo, and this gets loaded up into our JBrowse as another separate track for gene models. Okay, and the first bit of evidence we're going to go over is the intron splice junctions. So these are color-coded again. Say the red ones are on the reverse strand and the blue ones are on the forward strand. And it's quite good to remember this via red equals reverse, red equals right to left. That's how I always do it. And then the blue ones are forward. And so this is the directionality of the gene. So if you look here, the blue ones are going from left to right. Here we are. And the density of these introns is basically the evidence for that. So there's a lot more reads that kind of go from there to across the intron. That's what they're doing, the intron spanning reads. And so, once you've done a transcriptomic experiment, you basically are sequencing the mRNA, and this has the intron spliced out. So the reads that kind of cover this intron span, what is reflected here in this track. And if you click on them, as previously said, you get a little bit more information, so you can get the most abundant intron. Of course, this should be what's primarily represented in the gene model you're doing. But if you are creating alternate isoforms, you may want to look at different experiments and stages, because it can help elucidate different stage-specific introns. So if you look down here, tachyzoites, for example, if you want to do a tachyzoite-specific gene model for this gene, you may want to just utilize those introns that are highly represented within those samples. Okay, ooh, next one. So this is quite a new track. This is nanopore data, so long read assembled transcripts. And you may get this error message when you are looking at these genes, but if in the actual proper view, you get a nice kind of a transcript over that gene. So each one of these would represent, say, 5 nanopore reads, and they are kind of full-length transcripts. So this can give you a good idea of what is actually being produced from this cell or sample, because this is reflective of the mRNA. And of course, even though it's long read data, you may have kind of degradation of the mRNA beforehand in the library preparation or anything. So it's not 100% accurate, but it is very good, very good. Next, for this organism, Toxoplasma gondii, we actually have some computer-generated models using Craig, which is a program about 15 years old, I believe. And these were prepared over multiple different samples. And this just gives you an indication of what they predicted based on what was available at the time. And so these are quite, you could utilize these to generate your new gene models. And that's only kind of specific with Toxoplasma gondii. We don't have that for most of our species. Next one, chromatin modifications and epigenomics. So we have this data as ChIP data or ChIP-seq data, and this kind of reflects, depending on what histones you were looking at, and what kind of, whether you're looking at methylation or acetylation, whether this is indicative of activation and transcription, or whether it is about kind of just where chromatin has kind of non-gene expression, basically. So we have all this data in our website, and of course, as Omar elucidated earlier, just click on the select tracks, and you can look up. There's a whole section on this, and maybe read up about what they do, because each sample will be different. So whether it's a H3 K4 methylation or a H3 K9 acetylation, those kind of things, just double check what they can actually mean before going ahead and making your inferences. The combined RNA-seq, this is an amazing track. So this has taken away all of the sample data that we would have, and it's just combined them. So each one of these lines represents a different sample, and so you can get a good overall picture of the gene itself. And so as you can see in this gene, for example, maybe I'm not sure if you can see my cursor, but where you have a grainy background, this can indicate where transcripts have actually been extended over 2 exons. And so it's not 2 exons, it's just one big exon in certain transcripts. So it's kind of fuzziness. And of course, these peaks obviously are indicative of just major exons. So you have this kind of coverage, which is very nice. And in some arthropod species, you can actually see where the coverage, and you have introgenic genes, and you can demarcate them, if that's the word, from the enclosing gene. Other evidence types is, so back onto RNA-seq, this is our stranded data. So of course, I mentioned earlier, you've got reverse and you've got forward, so you can tell which genes are going which way. And of course, when this data kind of crosses over, you can actually see that in the gene models, and usually that would be like a UTR or something like that that's going over. But because it's stranded, you definitely know that that crossover is occurring. And this stranded data is definitely good for looking at different life cycle stages. So for example, this is just a quick diagram of T. gondii life cycle. So each one of these may have different gene expression patterns, say exon skipping in this life cycle stage, or not in this life cycle stage. So just to give some context. And then, of course, the reference sequence. So this is was mentioned by Gloria quite a lot. Not for these exercises, not that important, but previously, if you do see truncated genes that are, say, in one ortholog, it might be because we have incomplete genome assembly. So, some of our genomes may be a little bit old, and the assembly methods might be a little bit ropey, so not complete. And so you might have gene models that just stop randomly, like, okay, well, that's not going to be very good in a multiple sequence alignment because I don't have any other sequence. But this is just, you'll just see loads of ends, and this might be important if you are looking at certain genes that have this. Okay, the exercise. So for the next 40 minutes, you'll be put into breakout groups.